Good evening, ladies and gentlemen. Thanks very much for, it's great to see so many people here. We weren't sure how many people would come to Malaria Day. It's fantastic. And we want today to, because it is Malaria Day, to talk about the current malaria situation and to give a sort of snapshot of some of the work that goes on in Oxford and through Oxford on malaria. I'm just going to literally show a couple of slides at the beginning to orientators. I presume the fact that people are here, most people understand that malaria is a parasitic disease spread by mosquitoes. We often say it's a global problem, and it is a global problem, but that actually doesn't really quite get the picture, because if you take account of where the people are and the parasites are, this is what malaria looks like on a sort of on a map that takes account of the human population. Africa, a massive problem. Asia, particularly South Asia, a massive problem, and that's because obviously the population is so large. So you get a rather different, it is a global problem, but it's not an equally distributed global problem. Oops. One of the had we been having this meeting 15, 16 years ago, we'd have all been terribly depressed, and many of us were very depressed, and we were writing articles saying that malaria was a complete disaster, and it was a disaster. It was essentially out of control. Nobody had any real sense of what to do about it. There's massive problems of drug resistance. Mortality was very high. Nobody knew how many. We used to make up numbers. But it was and there was an absolute lack, apparently, of international or national will to do anything about it. Over the last 15 years, there's been a really remarkable change. I think a change which has surprised, more than pleasantly surprised many of us who work in malaria, because often people talk about sort of international initiatives and such like, and usually not very much comes of them. But in the case of malaria, there has in fact been a real revolution over the last 15 years. And it's partly reflected in this figure, which shows spending, global spending on malaria control. At the beginning of this century, the whole world spent only a couple $100 million a year on malaria control. And very quickly, after a series of concerted initiatives, this rose tenfold and has now gone up about 15-fold. So a really massive investment in malaria control around the world. You would hope that that would have some effect, and in fact, things have changed incredibly. Over the period 2000 to 2015, people are used to looking at these maps, or if they're not, you'll see a lot more, I think, from Katherine. But I mean, you get the general idea that sort of red's bad, blue's better. So you can see over a 15-year period, looking at Africa alone, there's been a major change, which you can just see without going down into detail, in the amount of malaria in Africa. But globally, over that period, over 600 million, it's estimated, over 600 million cases have been averted compared with the numbers in 2000. And really, I think extraordinarily, global malaria mortality rates have fallen by 60%, it's estimated, and in Africa probably somewhat more than that. Now, if you'd have asked any of us at the beginning of the century how likely this was, I don't think many of us would have put very much on this. So it looks as if everything's great, and things are good to a degree, but there are some big issues on the horizon. Firstly, drug resistance, and you're going to hear quite a lot about that and what both the situation and what can be done about it. Secondly, funding. It's fantastic that funding has gone up 15-fold, but it needs to go up 30-fold, and if it doesn't, we'll stall and things will return back to where they were. So things aren't totally rosy. In fact, we're at a very interesting point in history where there is many people are talking about elimination and even eradication. I think everyone should be aiming at eradication, but everyone should be more than that, aiming at being realistic. And unless we solve the problems of drug resistance, insecticide resistance, and funding, there is no point in talking about any of these things. And one of the key things we need are new interventions. So you'll hear quite a lot today about potential new interventions, both in terms of vaccines and also the power of genomic science to contribute to our understanding of malaria. So I'm going to introduce first Katherine Battle, who's going to talk about mapping. And I should have said that we talk about malaria as if it's a single thing. Of course, many of you will know it's not. There are several species. And in fact, we tend to talk about Africa, and we tend to talk about falciparum, because that's important. But in fact, there is another massively important malaria parasite. And just to ring the changes, Catherine's going to concentrate on that. So welcome, Katherine.
Hi there, thank you. Thank you, Kevin, for the introduction, and to WARN for organizing this event. I'm from the Malaria Atlas Project. We make pretty maps of where malaria is. And as Kevin said, I'm going to be concentrating on Plasmodium vivax, which has been my focus for the last several years, and how we have been mapping and addressing the challenge of this relatively neglected parasite. And because we have a strict 8 minutes to cover what we need to cover, I'm going to do a flying tour of some of the key points about Plasmodium vivax, malaria biology and epidemiology, why it's considered neglected or how, what challenges we've faced in trying to map this disease, and what we've done to try to meet that challenge. So as Kevin mentioned, there's 5 or 6, if you count the subspecies of ovale, parasites that affect humans for malaria. And the vast majority of the burden or global caseload is attributed to falciparum and vivax malaria. The biology of Plasmodium vivax does impart some unique epidemiological problems that make it difficult to control and even to understand in some ways. As shown here in this life cycle illustration of Plasmodium vivax, you'll notice that there's this liver stage. All Plasmodium parasites do enter the human liver at some point in their life cycle. But ovale and vivax have this ability to stay in the liver and form this dormant phase called the hypnozoite. That stage can then wake up at some point. It could be anywhere from a few weeks to months to years after the initial mosquito bite primary infection, and cause another blood stage infection and then another one after that. And this ability for it to sort of hide out in the liver allows it to overwinter seasonal transmission. It allows it to hide from blood stage treatment, diagnosis. And if we look at a map of what malaria looked like at the turn of the previous century, you see that it extended far into the temperate climate. And that would have been Plasmodium vivax, that light green way up north. And it remains the most geographically widespread of the human malaria parasites. It's still far up in North Korea. And an additional problem of this relapse, so not only does it hide out, but it's difficult to treat that liver stage. There's one currently licensed drug that can treat it, and it comes with its own problems, which could be a whole nother discussion on its own. But it's a long treatment schedule, which people don't like to take a drug for 2 weeks. And it can cause acute hemolysis in people who have an enzyme deficiency called G6PD. So some people are reluctant to give it as a drug. There's other things that Plasmodium vivax does that makes it hard to find. It circulates the blood in low parasite densities. It's often submicroscopic. It's often asymptomatic as well, so people don't necessarily have the classic malaria fever to go for treatment. So we have this widespread parasite. It's hard to understand. It's hard to find. So why would this be one that's considered neglected? And I've been told by a couple reviewers of papers that we can't really call vivax neglected anymore. The fact that I'm even invited to here to talk about it, I think, means that it's gotten some attention. But when we talk about it being neglected, it's relative to falciparum, because as Kevin said, when we talk about malaria, most people think falciparum, Africa. And just to kind of test this hypothesis, I did a search on PubMed of falciparum plus malaria, around 30,000 hits. Then vivax plus malaria, only around 7,000. So it is quite clear that there is more research output for falciparum malaria. And within MAP, our own database that we've assembled of cross-sectional surveys that look for infection in communities, we have Plasmodium vivax here. This is a map of all of our survey points. And we have around 13,000 points, which I was quite proud of because I was, one of the people going around basically begging people for data, and was feeling quite proud of that until I compare it to falciparum, which we have about 42,000 points for. So there's a huge discrepancy even within our own group where we use the same methods for both parasites that we don't have as much evidence to support what we try to do for both. So given what we have available and what we know about these parasites, what could we do in terms of trying to map it and understand how differences in the spatial distribution of this? So what we started doing at MAP was the first thing we did several years ago was we mapped the limits, the geographic limits of transmission. And that allowed us to assess what the population at risk is worldwide. And then within that, we estimated the prevalence using those survey points I showed on the previous slide. But what we're now trying to do is generate an estimate of how many new clinical cases happen each year, which is a very different metric, but we think is more operationally useful for people who work within country programs. So in order to go from prevalence to an incidence map, we first needed to understand, does relapse vary geographically? So this phenomenon of when the hypnozoite wakes up. Because there's been a long-standing hypothesis that the tropical strains will wake up quite quickly after the primary infection, whereas the temperate strains wake up more slowly so that they last through the non-transmission season. And so we did a large analysis and found that there are indeed geographic zones of relapse. So we developed a mathematical model to relate prevalence, which we had mapped, to incidence, which we want to map, and we had separate model fits for those different geographic zones of relapse, assuming that relapse would affect both prevalence and incidence measures. But our models and our outputs are only as good as the data that we have to put into that. So if you look on the previous slide, some of our relapse zones have almost no data, and some of the zones had no data. And I was fitting this to about 175 points. And these 13,000 points that I have as our evidence to predict prevalence, you can see that they're not homogeneously spread out throughout the vivax endemic world. We're heavily biased to areas that have national household surveys like DHS and MIS, which don't commonly search for vivax, and from areas where we have support from other Oxford institutions where we have strong collaboration. So by far, most of our data is from Indonesia, thanks to our colleagues based at the Eichmann Oxford unit. So what can we do to meet this challenge? We don't have a lot of data. We have a complicated parasite. And what we want to do is capitalize on the type of data that is strongest from different regions of the vivax endemic world. So in the Americas, they have strong, relatively strong health systems, health information systems. So we could use incidence data that is reported from health facilities to the government to measure incidence. And in other areas where we know that the government isn't reporting any vivax, like in Africa, we can use this modeled relationship that we have. And the outcome is that we have a hybrid map that utilizes health information system data, which we of course adjust for things like reporting completeness and treatment seeking, because we know that even in strong health information systems, not all the facilities report, and not everyone who gets sick goes for treatment. But even with those sort of flaws that we have to adjust for, we still think that an adjusted health information system database is better than a model that has a weak evidence base. As things progress towards elimination and strong control, the irony is that the less malaria an area has, the better that they are at finding their malaria cases. And we're finding that in some elimination, the countries that have already set elimination goals, that they're able to find, record, and geoposition cases better, such that we can make more fine-scale resolution maps and allow for malaria stratification for control and elimination subnationally. We only have a handful of countries, but I'm very optimistic that this is going to improve even further over the next 15 years, as Kevin showed with the funding slide. So that's a very brief overview of what we've done with Vivax and MAP, and I want to thank our whole team that's contributed in different ways, and our funders, the Wellcome Trust and Bill and Melinda Gates Foundation.
So we're going to move now to Philippe Guérin, and we're going to, he's going to introduce one of the big stumbling blocks that I've already mentioned. With this rather relatively rosy picture of progress, there's a real thing hanging over it, and that's drug resistance. And Philippe is going to introduce that whole area.
Mapping drug resistance. Well, what do we know about resistance? In 1957, after a few years where chloroquine was largely used and prescribed in Southeast Asia, we had a report of emergence of resistance. And you will see in blue in this video chloroquine resistance actually spreading, and then later on in purple SP resistance. So what happened was there were two foci of emergence of resistance in Southeast Asia and Latin America, and then a move westward, entering to the African continent and spreading all around the place. The same exactly happened 10-15 years later with SP, which was the alternative antimalarial, which was used starting in Southeast Asia, but the same phenomena of resistance spread was observed. Because chloroquine and SP was not working anymore in Southeast Asia, people were starting using mefloquine in monotherapy. And yet again, after a few years of use of mefloquine, emergence of resistance, making this drug totally useless in that particular region, and it was not even tried elsewhere. If we look at history, we can observe, I'm not sure if I can go there, we can observe a massive decline of malaria mortality, which started much before actually any intervention of drugs or insecticide, but a large decline of resistance. But then starting climbing again in the 70s, and that was mostly due to resistance. And the response to these first waves of the resistance, chloroquine, SP, and mefloquine, were slow. We did not properly assess the risk and the cost related to that, and resulted in millions of deaths, especially in Africa. Then we had the introduction of the artemisinin combination therapy. So trying to drop one of the drivers of resistance, which was use of a monotherapy, combining a treatment with different properties, and they were a great hope. And indeed, ACTs have been working very well and are still working very well. And that was what we were expecting, using ACTs with other intervention and seeing a large decline of mortality. Unfortunately, in 2007, the first confirmed report of artemisinin resistance was done by the group of MORU in the New England, and showing artemisinin resistance having emerged in the same region in the western part of Cambodia. What's the situation today? Well, it's reported to be confirmed in Cambodia, in Laos, in Thailand, in Vietnam, in South China, and in Myanmar. And so there have been a lot of debate most recently about is it a spread, is it an emergence? I think that's an interesting scientific question, but it's not really an interesting public health question, because whatever happens, it's likely that we will observe that. So it will emerge elsewhere or it will spread elsewhere, but it's unlikely that it will stay only in the Mekong region. And we need to collectively react about this threat, because basically we don't have an alternative ready now. There's not a new drug, which is not an artemisinin compound, available on the market. Just to highlight that there's already a few suspicion of artemisinin resistance, yet not confirmed in some places in Africa or in Latin America. This is not to scare people, but this is just to say that there's people already claiming that there's a problem with that. What we have done in WARN, we have tried to assemble data from different horizons, so clinical data, pharmacology, in vitro, molecular, medicine quality. And this has been a very large collaboration as of today of 260 institutions around the world who are collecting this kind of information. We are assembling this information, standardizing them, and this is resulting in having 400 clinical trials and more than 120,000 individual patient data in a repository which represent about 80% of all the published ACT data as of today. And this is a collaboration engaging also MalariaGen and MAP in this enterprise. What we have learned is that resistance is not an on-off button. Maintaining the usefulness of the current drug is really important. We can probably identify underlying factors promoting resistance, we can probably optimize the regimen that we are using today, and we can probably target the intervention where we should be focusing money and resources. And this is what we have done. So I don't have time to enter into details, but thanks to doing this pooling of all this data, we are able to identify particular risk factors associated with resistance. Some are the underlying factors that will actually facilitate the spread. So there's underdose in young children or in pregnant women, issues with comorbidity when a child is malnourished, issue with drug-drug interaction, poor quality medicine being massively reported in some countries, and a lot of regional diversity that we need to account for. We have done so far 22 individual patient data meta-analyses, and this has already impact in policies because this is providing information that can guide policy. So we have, thanks to this work, improved the dosing of the hydroartemisinin. We have demonstrated that the fixed dose combination formulation of the artesunate amodiaquine was much better than the loose dose. We are exploring optimized ACT regimen in malnourished children, and we have validated molecular markers being able to show the correlation with clinical phenotype. So we believe that this effort is actually possible by sharing individual patient data, which improve existing and future data, preserve efficacy of drugs which are currently used, and could be also lesson learned for the development of new drugs. You might have seen late last week there were a series of articles in Science talking about malaria wars. And one of the key sentences that I like to report in these interesting articles is that the science is uncertain, the data imperfect, and the tools far too crude. And we can only agree with that. There's a lot of data, but certainly not enough. So combating malaria through collaboration, this evening we had invited George, but unfortunately at the last minute he could not make it. But he sent us his advice for that. He said, well, combating malaria through collaboration, what else? So we think that really we have an opportunity here of making some changes by working together. And if we don't do that, I think that's going to be a collective mistake. Thank you.
It's a real pleasure to introduce Nick White. Nick White is, let's say, one of the longest standing and most notable Oxford malaria researchers.
Thank you, Kevin, for that most uncharacteristically generous introduction. So I'm going to continue the story that Philippe has just told you, and it'll be from the perspective of Southeast Asia. This is the entrance to a refugee camp, or a camp for displaced persons on the border between Thailand and Myanmar. And although we have a very small proportion overall of the world's malaria and of the world's malaria parasites, unfortunately, as Philippe has explained, drug resistance seems to keep coming from this region. And it's not once, not twice, but 3 times now. So the price in human lives of the spread of chloroquine resistance to Africa was literally millions of children died. That's not an exaggeration. That is the result of parasite genes that originated in Southeast Asia. And then, as Philippe has explained, the antifoal resistance, antifoals followed, they were the drugs that followed chloroquine, and they too came from this very place, Southeast Asia, in fact, from Western Cambodia. And now we have artemisinin resistance again coming from Western Cambodia. Now, if I can work out how to make this work. So this is Thailand is traditionally the graveyard for anti-malarial drugs. You see on the left, the left there, CQ is chloroquine, the last embers of chloroquine utility in the late 1970s, sulfadoxine pyrimethamine, which is an antifoal sulfur combination, following it and dying very, very quickly. And then a brief period when quinine, that's Q, was used before mefloquine was introduced in 1984, November. Actually, it was introduced in combination with SP, which was an interesting choice. Resistance happened very rapidly. We increased the dose. The dose was wrong. That's another repeated story through anti-malarial drug development, that we introduced drugs at the wrong dose. And so that we increased the dose, but that just provided temporary respite. And actually, by the late 1990s, we were really, really concerned that we would have untreatable malaria by the end of the millennium. And we were saved by these remarkable drugs which come from a traditional Chinese medicine called Jinghaosu or artemisinin, for which a Nobel Prize was awarded this year. And so in combination, not a particularly good idea to combine a dying drug with a new one, but that's all we had. So AM is artesunate mefloquine, and DP is dihydroartemisinin, which is another Jinghaosu-like drug, with another drug which the Chinese developed called piperequine. And both did really well for a very long time. But I'm afraid the recent story is not good. And the cupboard, the immediate cupboard, is bare. We don't have new drugs that we can take off the shelf and give to people. We do have, however, a rather healthy-looking drug pipeline, and I'll give you some good news. But before that, I would like just to emphasize how important the artemisinin drugs are, not just for the treatment of uncomplicated malaria, which fortunately is most malaria, but particularly valuable in the treatment of severe malaria. And they reduce the mortality between about 1/5 and 1/3, so a massive reduction in mortality in severe malaria. And we're very worried because of the nature of the resistance mechanism that we're going to lose that beneficial effect. This is the plant, and that's you'll immediately recognize that sesquiterpene lactone peroxide structure in the middle of it. Kevin's nodding. So that's the drug we use now. And that's the drug that it replaced quinine, which also actually comes from the bark of a tree from South America. And really, we don't have any alternative but to go back to that bark. So in Southeast Asia right now, if you have severe malaria, unfortunately, there are relatively few cases, we give both these drugs. So what's in the pipeline? Well, a few boring chemical structures. The first group of drugs are synthetic peroxides. So they've got that peroxide moiety, which are the things that look like a spectacle. But they're made, the one on the A is artemisinin, and B and C are synthetic peroxides. So we can make those entirely in the laboratory relatively cheaply. One of them, arterilane, is actually registered and used in India today. And artefenomel, or OZ439, is in development. It's still in phase 2. That means it hasn't gone through to the big trials that are needed to establish the safety and efficacy and get the drug registered. So some way off registration. But they represent an inexpensive approach, or relatively inexpensive approach. And it's unclear whether the artemisinin resistance mechanism will affect them. I personally think it will, so they probably are not going to be the answer to artemisinin resistance. Here's another very exciting, one of several very exciting compounds developed by the Novartis Institute for Tropical Diseases, which receives Wellcome Trust support, I should point out, based in Singapore. And this is a completely novel compound. It's called a spiroindolone. And it produces the fastest parasite clearance of any drug we've ever had. We thought the artemisinins were the best. That little cartoon on the bottom right shows the number of parasites in the whole body going down. And the red line is the results of the spiroindolone, cipargamin, and the green line next to it is the parasites coming down after artemisinin. So artemisinins, which are notable for being very rapidly and reliably effective, are beaten by this remarkable drug. This is also early in phase 2, and just to illustrate the uncertainty of drug development, it's hit a bit of a stumbling block because it has some toxicity. We're not sure if that's going to kill it or not. Very few drugs of those that are developed in the laboratory ever make it to human testing, and even when they get to human testing, not that many make it through to become a drug that you and I can take. Here's another drug, the one at the top left, from the same group. You'll recognize that as imidazolopiperidine. Kevin? Yep. Yeah. So that's another completely different compound, not like anything else, and it also, interestingly, kills the parasites in the liver before they grow into the blood, and it kills them in the blood, and it's rapidly acting, and it's well tolerated. But that also is early in phase 2. And finally, the other kid on the block is a drug which really looks a bit like chloroquine. The left part of the molecule is chloroquine, and then it's got this rather clunky side chain with an iron moiety in it, so it's called ferroquin. And that seems to be active against parasites which are resistant to chloroquine and all the other antimalarial drugs, and that's being developed by Sanofi. But what have we got today? Well, there are only two other drugs that aren't, or should I say, not widely used. One of them is atovaquone proguanil. Some of you might have taken that for prophylaxis. It's very well tolerated. It's not very rapidly acting, and resistance develops just like that. One single point mutation kills the drug. So when it has been deployed, as it was in Cambodia, I think misguidedly a couple of years ago, they had to withdraw it within 3 months. It was almost miraculous that it fell to resistance so quickly. And the other one is artesunate parinaridin, which is pretty good, but has not done well in Cambodia recently. So there's this gap, and what do we do? And this is the last slide. I think the options for the immediate future are pretty grim for us in Southeast Asia. And the most important thing, perhaps from a global perspective, is to make sure the problem stays in Southeast Asia. And that, I'm afraid, we have not managed to do yet. What can we do? Well, those are the only drugs we've got. We can give them for longer courses. That depends on the nature of the resistance mechanism. If it's partial resistance, then maybe longer courses or higher doses we can get away with. To be honest, higher doses are going to be difficult because these drugs are not that well tolerated when you increase the dose markedly. What we're trying is belatedly to combine drugs, even more drugs in the same combination. Now, when we treat TB or HIV or many cancers, we often use combinations of 3 or more drugs. So we're combining all the drugs we've got together. But as I've told you, they're all failing. So again, I'm not particularly optimistic, but the preliminary results are that we can get better efficacy at the moment. And one last point is a simple point, a public health point, that there's quite a lot of theoretical evidence that we should deploy different drugs at the same time with different mechanisms of action. That would slow the resistance down. But that's a sort of prophylactic policy for the rest of the world. But in Southeast Asia, right now, I'm afraid we're really up against it for the treatment of malaria. Thanks.
Great, thanks very much, Nick. I asked Nick before he talked whether he was going to be optimistic or pessimistic. He said pessimistic, but I think there was optimism in there, in that there are down the line some really promising drugs, and the real problem we face, as was obvious from Nick's talk, is the next few years, really. That really is very worrying. So we need new interventions, and we're going to move now to talk about potentially new interventions, and Sumi Biswas from the Jenner Institute is going to talk about malaria vaccines.
I guess you've just heard from Nick about resistance to antimalarials, which makes it even more urgent that we develop an effective vaccine against malaria. And developing a vaccine against malaria hasn't been easy. It is a master of disguise to our immune system. Parasite has adopted to spend its life cycle in 2 hosts, the human host and also in the mosquito. And even in the human host, if you look at the parasite, every time it comes out of an organ and moves into the other one, it actually changes its form. So our immune system is faced with multiple antigens of this parasite to deal with. And this also poses a challenge for vaccine development, because if you were to generate a subunit vaccine for malaria where you have to choose a crucial antigen to generate an immune response against, you're faced with choosing from 5,000 antigens which the malaria parasite has, but also choosing the right kind of immune system, immune response that you want to generate. For example, if you want to generate a vaccine against the sporozoites or the blood stage, you want to induce an antibody response, which are crucial for mopping up parasites. Whereas if you want to target the infected liver stage of the parasite is intracellular at this stage, and you want to develop a T-cell vaccine that would be crucial for eliminating liver stage parasites. In Oxford, we work on vaccines targeting all different stages of malaria, and our approach is to develop a subunit vaccine which will target the parasite at all these multiple stages. There are vaccine efforts elsewhere to work on whole parasite vaccines, and these are being developed in parallel. So our research in Oxford spans across right from the beginning, where we start with vaccine design. As I said, one could go for whole organism or subunit vaccines. And depending on the kind of immune response you want to generate, you want to choose your delivery platform. For example, from the recent Ebola outbreak, we know crippled adenoviruses and MVA can be used to represent an antigen to induce a T-cell response, and that would be very effective, for example, in the liver stage of malaria. Whereas for other stages where antibodies are important, like the blood stage and the sporozoites, we normally go for protein vaccines that you can administer with adjuvants, or virus-like particles, which are known to be very good at inducing an immune response. So once we've designed our vaccine, we test these in preclinical models to look if we are inducing the right kind of immune response. For malaria, we are able to culture the parasite for Plasmodium falciparum, so we can do assays where you can look if your antibodies are effective against a certain stage. Once these are promising, we then have a biomanufacturing facility in Oxford, very close to our research labs. And in this biomanufacturing facilities, these vaccines are produced according to good manufacturing practice, so they can be tested in humans. We are very fortunate in malaria research, for falciparum at least, to have a good model where we can test these vaccines. This is called the Controlled Human Malaria Challenge model. So for our phase 1 trials, after we have looked at the safety and the immunogenicity of a vaccine, we can then give malaria to these volunteers. They are closely monitored, and see if the vaccine or the effective immune response that we've tried to generate can protect against malaria. So I'm going to mention about one vaccine apart from the work in Oxford. There has been more than 30 malaria vaccines that have entered clinical testing. This is the only one that has entered phase 3 large-scale efficacy testing developed by GSK. And the vaccine targets the sporozoite stage of malaria. So this is the first stage, what the mosquito actually injects into us. And what GSK did was take their hepatitis B vaccine, so this is a virus-like particle of Hep B, and they presented a malaria antigen on the surface of the Hep B vaccine. And we know this vaccine is very good at inducing a high-level antibody response against the circumsporozoite antigen, and this antigen coats the surface of the sporozoite. And in the phase 3 trials being tested in more than 15,000 subjects, the vaccine has a modest efficacy. And we need to give 4 doses of the vaccine, and we know the vaccine efficacy wanes rapidly. It received a positive opinion from the European Medicines Agency, but the WHO committee has recommended to deploy it in a pilot deployment for 3 to 5 years only in a certain age group, the 5 to 17-month-old age group, because the vaccine efficacy was slightly higher in this group than the 6 to 12-week group that was also tested. So that's the most advanced vaccine currently we have. Professor Adrian's Hill's group in the Jenner Institute have designed a potentially improved version of this vaccine. So using current technology, he's been able to present more malaria antigens on the surface of this Hep B rather than more Hep B, and the idea being, hence you can induce a lot more antibodies. This is currently being tested in a phase 1 clinical trial in Oxford. And Adrian spent most of his life working on T-cell inducing vaccines, working on adeno and MVA. These are, again, the same vectors that was used currently in the Ebola trial. And he's worked on a target antigen called TRAP, which is expressed in the liver stage of the parasite. And very recently doing field studies with the KEMRI Wellcome Trust Unit in Kenya, this vaccine has shown modest efficacy over 2 months in a study in Kenyan adults. So R21 is the sporozoite targeting vaccines, and then this is the liver stage vaccine targeting the intercellular hepatocytic stage of malaria. And the idea being we can combine these vaccines together to have a better impact on malaria control. Simon Draper's group, again at the Jenner Institute, has worked on the blood stage of the parasite. So this is when the parasite invades new red blood cells and multiplies in the blood and is associated with most of the symptoms and complications of malaria. And vaccine against this stage is primarily to induce high antibodies against the antigens that are on the surface of the merozoite, so you block it from invading. So Simons Draper's group has worked on a very exciting new candidate called RH5. And the reason it's exciting is most blood stage antigens face with the problem of antigen polymorphism, because most of these antigens are under immune pressure. But this antigen seems to be relatively conserved. Simon's group worked with Matt Higgins to solve the structure of this protein, and he's shown in non-human primate models that you can get heterologous protection against different malaria parasites using this antigen. Again, this vaccine has been produced to GMP and is going to enter clinical trials in 2016. Lastly, the sexual stage vaccines. These are different from the traditional vaccine, in which case we are trying to induce antibodies in humans, but these antibodies are going to be effective in the mosquito, because the antigens against which the antibodies are raised are actually expressed only when the parasite is in the mosquito midgut. So the idea being, when the mosquito takes up a blood meal, antibodies will prevent the development of the parasite in the mosquito midgut, and the mosquito won't be able to transmit the parasite. Again, we have leading approaches for this in using an antigen called PFS25, and we have funding to test a nanoparticle vaccine, a virus-like particle expressing this antigen in phase 1 trial, again with our collaborators in the Khemri Khalifi Research Institute funded by the Wellcome Trust. So our approach in Oxford has been to target the parasite by developing vaccines against multiple stages. These work by different mechanisms and different kind of immune response, and the aim is, by combining effective vaccines against all these different stages, we will be able to come up with a deployable vaccine for the future. I have not mentioned vivax. We do have an active program on vivax vaccine, and following again the same principle of targeting the parasite from with different stage vaccines against vivax malaria. Thank you.
Great, thanks very much, Sumi. So our final speaker before we go into the open discussion session is Dominic Kwiatkowski. And one of the real sort of revolutions that's happened in all science, I guess, but I mean, I suppose from where we stand in malaria, our interest is what it's done to malaria, has been the whole change that genomic science has brought right across the board, from from epidemiology right through to product development. And one of the leading lights internationally in that whole area has been Dominic, and that's what he's going to talk about this evening.
Well, thanks very much, Kevin. We've had a great talk from Katherine about mapping malaria and why it's so important. But here's a catch. Our current maps of malaria are based mostly on measurements of how many people are infected. And there remain large knowledge gaps about what's going on in the parasite population. For example, to what extent are parasites flowing between different locations? How much does the parasite population in a village change from one transmission season to the next? And local resurgences of malaria are often unexplained. And these are important questions if you're trying to control malaria. And one way of getting at them is through genomics. Nick and Philippe have talked about Southeast Asia being the global epicenter of drug resistance. So let's focus our discussions on what's going on there. Over the last decade or so, artemisinin resistance has been spreading across the region, as Nick and Philippe have said. That's because of a mutation in a particular gene. Researchers were quite quick to get onto that compared to previous episodes of antimalarial drug resistance, because they had genomic tools, and those genomic tools allowed us to localize the position in the genome where those mutations responsible for artemisinin resistance live. So for those who aren't familiar with genomics, the parasite genome is a book comprising about 23 million letters. It's rather boring repetition of A, T's, G's and C's. It's got 14 chapters called chromosomes, and those encode these 5,000 different genes. And artemisinin resistance is caused by mutations of a particular gene that was previously people hadn't really paid any attention to, called KELCH13. Now it turns out that as we analyze the genome sequences of those parasites, it isn't just a single mutation in KELCH13 that's spreading across Southeast Asia. Rather, what's happening is that independent mutations are popping up in different places. And we know they're different for two reasons. Sometimes they're in different parts of the gene, but they still have this effect on resistance. And sometimes they're in exactly the same position in the gene. So that position in the gene looks the same between different places. But by looking at the surrounding positions, we can see that the mutation has risen independently on a different genetic background. Now, that's what sort of genomics has helped us do over the last 5 years or so. What interests us going forward over the next 5 years as genome sequencing becomes more commonplace is to analyze the problem at a different level. Let me just first of all talk a bit about what the revolution that's happening in the technology. When the first parasite genome was sequenced, I don't know the exact cost, but I think it was upwards of £20 million. And these days we can sequence a parasite genome for on the order of £20 as a marginal cost. Previously, it required large samples of blood, and now we can do it often on just a single dried blood spot. Previously, it required a lot of parasites to be in that blood, and we're driving the level of parasites down. Right now, we still require the individual to have symptomatic malaria and a fair few parasites in the blood, but over the next 5 to 10 years, that level of detection will progressively get better. And what the information we're getting out of that is getting more complete. With our current technologies, we can find vastly more than we ever could before, but over the next 5 years, we'll start to see the full genome sequence. Right now, parts of it are obscure to us because they're so variable. So the technology is moving fast, and it's going to get increasingly implemented in epidemiological studies. And so if we're trying to do something about the spread of artemisinin resistance, and obviously the concern is to stop it spreading outside the region, but also to attempt to eliminate malaria from within the region, we need to know things about how parasites and how resistance is spreading. And what we can see already, just from the existing level of genetic data, is that the parasites live in what we call tribal groups. Tribal groups are groups of individuals who share ancestry because they mix and breed with each other. And by defining the parasite tribes, just like defining human tribes, we can start to get a picture of how things are segregated. We can do that increasingly as we get more genomic data because every individual is a mosaic of many ancestral genomes. Parasites are sexually recombining with every transmission cycle. And by looking mathematically at those patterns of ancestral sharing across the genome, we can group parasites into the tribal origins. And a tribe, just like a tribe of parasites, just like a tribe of humans, has individuals that are quite different from each other, but they have some common features that make them different from other tribes. And often, there's only one tribe in a given location, but tribes are maintained by patterns of breeding. So just as in human populations where you may have different tribes because they may have different cultures or different religions or whatever, in a parasite population you may have different tribes because they go through a different mosquito species. So what we're interested in doing is getting high-resolution sampling, high-resolution genomic data, and these are simulations produced by Jacob Almaguer. And what he's showing is our idea of what sort of things we'll be able to do as we get denser sampling and more complete genomic information, taking locations and being able to sort of map out the spread of a particular tribe and then combining this into a map where we can see in a country how different tribes are intermingling, getting a sense of the flow of parasites, getting a sense of if we were to, for example, try to eliminate malaria from this region here, would it immediately come flooding back in from this region here, or is there enough of a gap to allow us a bit of time to do each region in sequence? So that's what we'd like to do. My thanks to our team at Oxford University and the Sanger Institute. Thank you to Jacob for the slides. And if you want to know more about our work, I suggest you go to the MalariaGen website, Google on MalariaGen, or there's the URL. And thanks very much to Rachel Giacomo-Antonio for the wonderful new website we've got.
Well, that's great. Thanks, Dominic. And thanks to all the speakers for fantastic talks delivered absolutely on time, which allows us to do exactly what this meeting was about, which was to open it up for discussion to the audience for half an hour on anything you've heard or even stuff you haven't heard. I'd like to invite the speakers to come to the front, please, and sit at the front. Okay, thanks very much. So I'm going to turn that over to the panel. I don't know who wants to take the first one. The first question was about interactions between falciparum and vivax, and one could either talk about that, biologically, geographically, treatment-wise, any way one wanted. So, Katherine, I don't know if you want to go first with that?
Sure. Of course. Is this close enough to hear?
I think so. Let us know if you can't hear. It sounds to me as if it's good, yeah?
So as far as mapping the two parasites, we've kept them very much separate just because of the methods that we use have been different so far. But it's definitely something that as we get into this kind of fine scale mapping that I mentioned on my very last slide, it's becoming more important. But what we've seen as far as our data that's available is that where they're co-endemic, vivax doesn't tend to get picked up as often. So there's some diagnostic biases to it, but then there's also some of the biology where, and then I'm probably not the best to speak to this, but where it's simply not detected, even if it is there. So it's something that we're considering how best to represent this in our maps to show, especially because of the importance of mixed infections potentially on clinical outcomes. But as of yet, that's something that we're still working on as far as how to find these mixed infections and how to show them in the maps and how important they are.
Great. Nick, do you want to, or does anyone else want to comment? I wonder if you want to comment on the biology, as it were, the interactions within a single individual, some of the sort of stuff that you've been considering.
Okay, so these two parasites are actually in competition with each other, and we know that because in the good old days when we used to treat neurosyphilis with malaria by giving malaria to people, people actually did that. They gave the two together, and you could see that one would suppress the other, and usually it's the muscular falciparum that pushes down the rather effete vivax. I actually think relapse may be a strategy to avoid falciparum, but in Southeast Asia, where Katherine has shown you you have very frequent relapses, the two commonly coexist, and interestingly, falciparum malaria is an important cause of vivax malaria. How does that work? Well, about 1/3 of people who get acute falciparum malaria will have a relapse of vivax afterwards. So that, if you add that up, that means that falciparum as a stimulus, if you like, to vivax relapse becomes very important. So these two are coexisting and trying to avoid each other, and as Katherine points out again, vivax tends to be underestimated. So as falciparum comes down, you start to see vivax. It's always been there, but you underestimated it initially.
So the next question was about chloroquine resistance in vivax. Perhaps you'd like to start it, then maybe Philippe fill in if you leave anything out.
So yeah, chloroquine resistance is very interesting. We haven't actually nailed chloroquine resistance. We don't understand the molecular mechanism as we do for falciparum malaria. It's insidiously spreading. You get high level chloroquine resistance in vivax in Indonesia, on the island of Sumatra, and throughout the Indonesian archipelago, and particularly on the island of New Guinea, so Papua New Guinea and West Papua. Elsewhere, it's low-grade resistance, but it's increasing late failure rates. In fact, they're probably failure to suppress the first relapse. As a result of that, the latest WHO guidelines do recommend now ACTs as an alternative, and that allows it's very good. It allows you to have a unified treatment policy, and so ACTs are recommended. One caveat: vivax develops resistance to pyrimethamine very rapidly, so not sulfadoxine pyrimethamine, but all the other ACTs can be used, and so probably chloroquine is gradually going to phase out.
Philippe, did he miss anything out?
Well, maybe just a comment. There's a lot less data on vivax than on falciparum, and we are starting an effort of pulling data on vivax, and this is a work which is led by Rick Price. And this is just the beginning of that. But when we look at the data from what we have seen already, the challenge of assessing this data is going to be higher than on falciparum. So vivax is a complex beast, and it's going to take probably more research and more data collection to get to the understanding of that. One last thing that we are observing in Africa, for instance, like in Angola, we have seen a massive increase, and in Ethiopia, a massive increase of vivax in proportion of falciparum in the last decade. As we see falciparum declining, we see the proportion of vivax increasing. And so this problem, which was sort of only thought to be in Asia and Latin America, is likely to be also a problem in Africa at big scales in not too long.
Okay, so anyone else want to comment on the vivax-falciparum interaction? If not, let's move to the third question, which was what's driving resistance from that one small part of Southeast Asia? Dominic, do you want to have a go at that? I mean, I've heard you venture views on this.
Well, I don't know.
Yes, that was the view you ventured last time, yeah.
I mean, I'm not necessarily the best to express all the vast range of different views that have been put forward. But clearly, some of it is possibly to do with history of drug use, and these drugs were introduced early on, and there were some rather dubious policies for administering antimalarial drugs over the years, so there may be partly historical issues. Partly it's to do with patterns of drug use. There's a lot of drugs being used, but also it's a region of relatively low transmission compared to Africa. And that means to say that many cases are symptomatic and many cases get treatment. Whereas in Africa, of course, many people are asymptomatic and never get treatment. So if you're in the parasite population, mostly in Africa, you're not getting hit by drugs. Whereas if you're a parasite in Southeast Asia, if you think of it as a game of whack-a-mole, the parasite's getting whacked more often in Southeast Asia. So that could be another factor. It's also plain that the parasites, these tribal groups that I talked about, have got some very interesting features about them, and it looks like maybe some of those features make it easier for resistance to evolve in those particular tribes. But I'm sure that Nick's got lots of other possible reasons.
I'd just like to say that in the large trials that have been conducted to map artemisian resistance, led by Dr. Liz Ashley, who's hiding in the corner there, the parasites were made available to anybody who wished, any bona fide investigator who wished to investigate them. So there are a lot of artemisian resistant, well-characterized parasites available for researchers should they want them.
Yeah, thanks. Well, while people are still cogitating about it, I'm going to ask Dominic to start on that, and then anybody else who wants to come in, because, well, simply because I know some of the work that's going on in Dominic's group.
Well, as you know, malaria is transmitted by a whole range of different Anopheles species. And as you know, in Africa it's dominated by some major species groups, Anopheles gambiae complex, for example, Anopheles fenestus. And as you know, in Africa, they tend to bite indoors in general. And so it's turned out that vector control in Africa has been a tractable problem. And now Nick, I know, can speak to this much better than I can, but in Southeast Asia the problem is there's a much greater diversity of vectors. They're often biting outdoors. They often, in a village there'll be many Anopheles species and no one's quite clear which are transmitting malaria, and they may be transmitting it in the jungle, etc. So vector control, relatively speaking, has been less of a powerful tool in Southeast Asia. Is that fair to say?
Absolutely.
So that's really the big that's the big difference. That's the sort of big thing that's going on in the vector. Now, and as you know, in the maps that we saw of the control of malaria that's going on in Africa right now, the vast, most important thing in reducing malaria mortality in Africa over the last 15 years has been the use of insecticides, insecticide-treated bed nets, indoor residual spraying, and as you know, insecticide resistance is now sweeping across Africa. So what's going to happen there is unclear. There's a lot of talk about using gene drive systems, etc., potentially new insecticides, but many of the issues that we've heard talked about in relation to drug resistance and vaccine development apply also to the mosquitoes. Is it going to work? Is it going to be sustainable? How do we guard against new forms of resistance? So there's a huge host of problems there, and obviously we think, since from a genomic perspective, that can somehow help in classifying some of these problems, but they're very practical problems in the field.
Do you want to say, you mentioned gene drive, do you want to say just a little bit about that? Just people may not be so aware of the kind of sort of wild ideas that people are playing with now.
Well, it's been speculated for now for quite a long time that there could be genetically modified mosquitoes, and that could work in a number of different ways. The field has really sort of taken off. Everyone's got very excited about it in the last year or two because of a very efficient system for modifying genomes known as CRISPR-Cas. It allows you to edit genomes extremely effectively, and now everyone is thinking that this will be the way forward for modifying mosquitoes. On the other hand, it turns out we've been genome sequencing mosquitoes across Africa, and it turns out that thus far in Anopheles gambiae we find one variation every 2 bases. So firstly, finding a sequence that's sufficiently constant to get a gene drive system in, there's a limited number of sites you can target, and there's the potential that the mosquito population will adapt. So there's probably no magic bullets, but there's a lot of excitement about potential new approaches.
First question about whether, a mixed question, ACT resistance less likely in Africa because of the relative less use of monotherapy, and also some more consideration to the idea of triple therapy, which Nick mentioned in his talk. Nick, I guess we should put this one to you.
Yeah, I think there's several reasons why artemisinin resistance emergence would be less likely in Africa, but unfortunately it hasn't been possible to prevent monotherapies being used, despite WHO's, I think, quite forward-looking and brave ban. It was many years ago that the former head of the Global Malaria Program, Arata Kochi, said ban monotherapies. But unfortunately it hasn't been possible to really make it work everywhere. Still, I think for that, because of the higher transmission setting, it is probably less likely. The more difficult one, perhaps Kevin would like to comment, is whether we can persuade people generally to use 3 drugs, which means more adverse effects and more costs. We managed to do it for TB, we managed to do it for HIV, but malaria is a remarkably conservative grouping, and I don't know whether we can in malaria.
Anybody else want to comment on it? Yes, Philippe. And then maybe also slip into the second question as well, was whether we can get to near elimination without resistance. I guess sort of finally, it's almost like an arms race.
Well, first a comment on monotherapy and the quality of antimalarials also circulating in Africa. That's a piece of work that we are doing in WARN and led by Paul Newton. And we have been observing a very, very large amount of poor quality medicine, not only falsified or counterfeit medicine, but poor quality medicine circulating in many endemic regions, in Asia, in Africa, and in Latin America. We don't quantify that very well because there's very scarce data, but each time that it has been looked at it seriously, that's really striking a level of poor quality medicine circulating, and that's exactly what you would do in a lab. You give a subtherapeutic level of a treatment, and then you get resistance. And this is happening. This has happened, and this is happening today in Africa because of a lack of a capacity to really measure the quality of medicine. And despite the ban of artemisinin in Africa, and this has been working progressively better. There's a lot of poor quality ACT circulating in Africa, and we don't really talk about that. It's a sensitive subject, difficult to measure, but also sensitive because this is also touching the generic companies, and we don't want to affect the huge impact that has been in access of the generic companies. But on the other hand, we don't tackle properly, I think, at this point, poor quality medicine.
Great, thanks. So let's take, and does anyone want to comment on that question, which I took to mean there being something of an arms race, as it were, but can you get to elimination without resistance just becoming the absolute, barrier to it? Philippe, do you want to comment on that?
Yes, if you're quick. I'm not sure I want to comment on that. Elimination without resistance. Well, there have been some good results already in some part of Africa where we see a massive decrease of cases of symptomatic malaria. Now, are we really effective? I'm not sure. When we are looking now with much more sensitive methods than microscopy, like PCR techniques, and we see very striking level of parasitemia in patients in, let's say, Dakar, in an urban area where supposedly there's no transmission. Well, we find 20% of people having some parasites circulating in their blood. And so I think unless we have proper methods and proper ways of getting to this reservoir, I think we are a long way away from elimination.
Nick?
I think it's a matter of how quickly and effectively you do it. If you do it very quickly and very effectively, yes. And that's why we've been making such a fuss about artemisinin resistance. We know that the longer you drag it out, the more selective pressure you're providing and the higher grade resistance will happen and the partner drugs will fall like dominoes, and that's exactly, I'm afraid, what happened. I mean, we've known about artemisinin resistance in 2007. So Sri Lanka, for example, is eliminated now. They've eliminated without resistance.
Okay, let's just take the final question, then we'll take some more questions from the audience. And this was the effect of climate change, the potential effect of climate change. Will we be able to start doing our malaria research in Holland and the Fens again, as we used to be able to do? Katherine, I guess this falls more naturally in the mapping group, and certainly some of your colleagues have speculated on this.
Yeah, this is something that we've looked at in MAP with how we've looked at trends over time. And the change in climate that we've seen happening that could drive up malaria is luckily not as quick or effective as all the efforts that are going in currently to control malaria. So our control is more effective than climate change. I mean, there still are some epidemics in a warm period in, say, the Kenyan highlands that happened in the late 1990s. But overall, we're doing a better job at controlling it. What I think is probably going to be more of a problem than climate change is changes in land use. And this is what we see a lot in Asia, again, in this problem area where we see resistance, is we also see deforestation, which creates different environments and can allow breeding sites and changes in transmission. And I think that's kind of the thing to keep an eye on, is how we're using and changing the land.
While we are passing the mic, I'd like to comment on the AMFM. An interesting outcome of IMFM, which has not been really noticed. One of our colleagues at the London School of Hygiene and Tropical Medicine did a very interesting assessment of the quality of medicine in Nigeria. And they found in one of the states 13%, 13% of poor quality medicine and almost no falsified medicine in their very good survey in 2000, which was conducted in 2010-11. And we were sort of very surprised by that, and they say, well, maybe the problem of poor quality medicine is not that big in Nigeria. And when they presented these results, there was somebody from the Global Fund who was part of the AMFM effort at the time, and she said something very interesting. She said, well, that was also the year where AMFM was scaling up in Nigeria, and we were pouring Nigeria of good quality medicine. And I think that was a very, actually, good demonstration that if you made available good quality medicine, you pushed away poor quality medicine and falsified medicine.
No, it's an important point, although correlation isn't causation, as we're often reminded, but well, it is because it is just undoing sort of all your sort of lecturing to students. Sumi, you've been sitting patiently waiting for a vaccine question. Here's the big moment.
I think firstly I would like to say it is a very important milestone, the efficacy results of RTSS. It's a vaccine against a parasitic disease that has shown any efficacy, so that is good. But as you point out, the efficacy is very modest, And there are all the likely implications of short-term efficacy, which will probably shift the malaria incidence into older population. And it is effective, and a lot of mathematical models have suggested it is cost-effective and it averts case, etc. But I think what we're currently doing will be very informative. That is to deploy it, first of all, to see logistically if it is possible to deploy this vaccine, because it requires 4 doses as opposed to 3, which we can fit into the extended program of immunization, but also looking at a lot of other indications over time whether this vaccine will be effective in controlling malaria. So as you raised, I think these are things that we definitely need to look for and measure, but also keeping in mind that it has conferred some protection against a parasite that has been very difficult to achieve.
Okay, great. So as we enter that sort of important few minutes before we all go out for the reception, I want to just ask each member of the panel in 30 seconds or so to comment on the fact that in the Twitter sphere today, those of Nick, I suppose you've been on it all day, there's an enormous amount of sort of naive optimism about eradication and such like. We all want to eradicate malaria. But I want to ask each panel member to say how, on Malaria Day, when the sort of the word has been ending malaria, how optimistic are you that malaria can be ended in that sort of relatively short time span that people are talking about? And what would make you more optimistic? What one change would make you more optimistic? So we don't need a big discussion, just want to know, are you optimistic or not? But what I really want to know is what one change would make you more optimistic? Dominic.
Oh. Well, I mean, I think it's a disease that can be eradicated, and I know it won't get eradicated unless people want to eradicate it, so setting a timeline is good as long as it's not detrimental to other things. So I'm completely behind it, and whether it's 25 years or 50 years or whatever, we have to have a timeline and a roadmap. We have to be realistic. I think that the one thing would be realism.
Okay.
Just,
Yeah, that's great. Philippe, optimistic or not, and what one thing?
Well, I think we need to learn at polio, possibly as an example, and we see how painful it has been to get where we are on polio. I think it's unlikely that the world will go back to another exercise like that, and we will need probably to integrate, so that's the positive note, we'll need to integrate the malaria effort with other efforts that are in public health that are facing those countries. Unless we do that, I think this is going to be a waste of time.
Okay. Nick, you optimistic?
I was optimistic about 10 years ago. I've become less optimistic. I agree with Dominic. I would say honesty. If there was actual honesty, true honesty in the dialogue, so that we did talk about things like corruption and bureaucracy and ineffective national malaria control programs, things we're not allowed to say, then I think we would have a better chance.
Okay. Sumi.
Yes, I am optimistic, and the one thing that would make a difference, and I'm partially biased, is an effective vaccine, and I think that has to be used with all the other tools that we currently have, and one day we will get there.
Great. Finally, Katherine.
Cautiously optimistic, I would say. I think when we talk about eradication and getting rid of all malarias, when we consider things like nosy, that's where we have to be cautious. But I think when we look at the progress that we've made with PF and PV, that this is a possibility. And my one thing would definitely be a vivax planted, a safe, effective treatment for the hypnozoite that people want to take. I think could get rid of this long-standing parasite of PV.